Well, good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Few-Shot Prediction of Drug Target Binding Affinities for Small Molecule Drug Discovery. My name is Ryan Muse, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speaker. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, please contact me at any time by sending a message using this same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Standigm, who developed the content for this presentation. Standigm is a workflow AI-driven drug discovery company headquartered in Seoul, South Korea, and subsidized in the US and the UK. Standigm has proprietary AI platforms encompassing novel target identification to compound design, all to generate commercially viable drug pipelines. Pursuing full-stack AI-driven industrializing drug discovery, Standigm has achieved the automation of molecular design workflow, and the automation effort has been expanding to the whole drug discovery process based on Standigm AI platforms, including the Standigm Ask for novel target discovery and Standigm-Best for novel compound generation. Now I would like to introduce our speaker for today's event. As an AI director of Standigm, Jiho Yoo, PhD, is focusing on the development of Standigm's AI module technologies. Prior to Standigm, Jiho developed AI algorithms related to organic materials and automatic synthesis at Samsung Advanced Institute of Technology, SAIT. He received a PhD in computer science, machine learning from Pohang University of Science and Technology. He is a co-author of several publications about molecular design and machine learning. But now, without further ado, let's go ahead and hand things over to our speaker for today, Jiho Yoo. You may begin when you're ready.
Okay, thank you, Ryan. And hello, everyone. Thanks for joining this webinar. Okay, so this webinar is about data-driven machine learning method for binding affinity prediction. The amount and quality of data is always important to build accurate machine learning models. Nowadays, the deep learning models for AI are really large and have huge number of parameters. So usually the scale of data becomes more and more important. But in many tasks in drug discovery, data is not sufficient. So we only have a small number of data for certain tasks, mainly because the experimenter itself is quite difficult and expensive. And also because the target system is quite new and has not been studied much. So in fact, there are some companies at scale generating large amounts of data, for example, through phenotypic screenings and analyzing the data with AI-based methods. But it requires a large amount of, actually huge amount of investment, and still there are remaining cases not covered by the large-scale experimental models. So if there are only a small number of data points available, a few-shot learning is one formulation of such problems. It aims to build an accurate model with only a few data samples given. So meta-learning is an approach used in deep learning community to deal with challenging practical tasks, including few-shot learning problems. So in this talk, I will explain about a novel few-shot meta-learning model, which we recently proposed. Okay, in this talk, first I'll explain about the backgrounds and the problem we are focusing on. We have examined a few strategies for the problem, so I'll describe about them, like a little bit chronological way. So along with our final model, you will see some of our previous attempts. And then I'll explain about the main algorithm, which we call it as meta-DTA. So the name metality comes from the fact that it is based on meta-learning method, especially for the problem of few-shot learning. So I'll explain about the concepts of meta-learning and few-shot learning, and then the model itself and the results will be described. Okay, before I start, I'll introduce our company Standigm again. So Standigm is an AI-based drug discovery company founded in 2015. So now we have about 80 employees having different specialties, ranging from developing AI models, managing data and workflows, and modeling and analyzing with computational and medicinal chemistry and biology, including systems biology. So we are focusing on developing AI-based methods and applying them to early drug discovery. And we have been building the AI-based platforms introduced before by Ryan to provide standardized and automated workflow for drug discovery. So two main platforms of Standigm are Standigm-esque for target discovery and Standigm-based for drug discovery. So for a disease, you can ask which target is best for remedying the disease and then find best compounds for candidates for next stage. Okay, and today's topic is related to the best platform for drug discovery process. So beginning with hit identification and covering through the lead optimization. So the best platform aims to discover novel preclinical candidates for given targets. And the best platform consists of various sub-modules to deal with diverse tasks during the process. For example, there are modules for data management, model training and inference, and the computational research management, and analyzing and summarizing the obtained data. And the modules for more specific downstream tasks, such as prediction of compound properties, such as primary activity and selectivity and so on, and screening compound libraries and designing optimized molecules, molecular structures. So the model I'll explain today is mainly built for primary activity prediction, so which is the most essential characteristic of the compound and should be maintained and optimized further during the drug discovery process. So for the primary activity prediction, actually the most systematic way to discover active molecules, I think, is the high throughput screening. So, however, it is quite expensive and so risky for being used routinely in small scale companies like us. So instead, we are focusing on more cost-efficient computational methods. So structure-based and ligand-based methods are two most widely used computational approaches for finding active molecules. So structure-based method tries to predict actual interaction between the target protein and the drug compound, mainly through the docking simulation, which predict combined structure of a drug and a target and calculating the affinities of binding is in the form of the docking score. On the other hand, the ligand-based method starts from known active compounds and tries to find compounds similar to them. So usually by comparing the shapes and the pharmacophore distributions of the compounds. So for successful application of these approaches, structure-based method actually requires an accurate protein structure model to actually improve the accuracies of docking simulations. And ligand-based method requires known active compounds. So active compounds have to be known a priori. However, we are also dealing with target discovery, and finding many novel targets which does not investigate much yet. So for those cases, the information required for running structure-based or ligand-based methods are not available. So our main objective is set for those cases. So So this is categories of the cases. So if we have structural information, so we can simply apply the structure-based method. And if we have active ligand information, we can apply ligand-based method. So if we have both, we can also apply both methods and combine the results through, for example, like applying the consensus rankings or so on. But we want to build a method for the remaining case, so where the structural model or active compounds are not known yet. So in other words, we want to build a systematic way to start a drug discovery from the scratch, so without running experiments requiring too much cost. And we thought that AI-based methods could be helpful for this case in some way. So we can think of the approaches. So we can think of the various approaches for this case. So the first possible approach is focusing on building of the protein structural models, so push forward for structure-based methods. So if we figure out the accurate protein structures in a systematic way, it will be good to be used by the computation method. We can apply the structure-based method with the virtual protein structures. And as we know, AlphaFold 2 and the following models recently has shown astonishing accuracy for protein structure prediction. However, after the protein structure itself is revealed, there are many remaining things which should be modeled to complete the protein structural model for docking simulation, such as binding site information and structural changes of protein induced by the binding processes. So usually, revealing it requires human investigation and modeling. So we think that it is not easy to use this approach in a systematic and scalable way. So we want to build a platform for the process. So the second one is try to build structure-free activity prediction models. The idea behind it follows a philosophy of deep learning, where the methods for intermediate feature extractions and processing are determined automatically during the training of the model with large amount of data. So in many cases, the trained models have shown better performance than the models based on manually designed intermediate features. So the structure-free model takes low-level protein features, such as protein one-dimensional sequence information, like AlphaFold, and then tries to directly predict the activities of a given compounds for target. So the process of three-dimensional structural interactions are intrinsically modeled in this kind of models. And recently, many methods following this approach have been proposed, and they have shown quite promising accuracies for predicting drug target interactions and binding affinities. However, we believe that this kind of huge and complex learning model still requires much more data to be properly trained. And there are some of the interpretation of these model results, which indicating that the model does not exploit the proper part of the protein sequence to make the predictions, which is, for example, the part of the sequence located really far from its binding sites. So we thought that this is not our approach for now. And the third one is more like a direction of the ligand-based method. And the conventional ligand-based method is based on the known active compound. So we can call it as active ligand-based method, so which focuses on a small number of available highly active compounds. Because at the very first stages, these highly active compounds are not available. So instead, we thought of an AI model which predicts the compound activities by using all available known activities of the compound data. So if sufficient amount of activity data is given, the model may figure out the interaction patterns of the target protein from it. Or more directly, the model could screen out the inactive compounds using the information about the known inactive compounds, or combine the information of compounds having some minor activities to suggest the compounds with stronger activities. So we hope that the activities like that. Then we can call it this kind of approach as a ligand activity-based method to distinguish it from the conventional active ligand-based methods. Okay, to be precise, to summarize, and our approach can be called a ligand activity-based method to lower the hurdle for binding high activity compounds at the very first stage. Instead, we think of gathering and using all available activity data for compounds having little or no activities. So our AI model will take the whole available activity data as input and predict the activities of given test compounds. So there are two different ways we can think of to use the compound activities. So one is the global type and the other is the local type. So in the global type scenario, the activity data is used as the kind of target specifying features. So like three-dimensional structures for the structure-based models, and one-dimensional protein sequences for structure-free methods. So we use the data set as actually some descriptors for the target. The set of compound activities are used to determine the characteristics of target proteins. And the other one, the local type scenario. So each activity data is used to determine the activities of the query compound as a form of the reference point. So it is a reference to calculate predict the activities of the query compound. So it is more like the ligand-based prediction of the activities, where mostly the activities of similar ligands are used. So in other words, each compound activity is used as a reference points. Okay, so we also design a workflow for applying such AI model to be applied for the very first stage of the novel target drug discovery. So first, we have to perform the initial experiments on a small number of compounds and obtain the activity patterns of them. Then an AI model is built with available external data and the newly obtained activity data for specific target. And by using the trained model, we can screen the compound libraries to find novel active compounds. So this will be our workflow from the scratch. So I think at this point, it may be a little bit unclear what the model is actually doing. So this is an illustrative explanation. So we can think of an image reconstruction problem where only a small number of pixels are given. So the AI model in this case tries to reconstruct the original images. So if the model is properly trained with a large amount of relevant image data, the model can reconstruct the original image in a quite similar way. So it's all the faces here. And you can see that in this example, the model can reconstruct at least a similar image with less than about 1% of given pixels. So really small amount of data is used to reconstruct this image. And similar to this, and the image becomes more and more closer to the original image when the number of given pixels increases. Similar to this, we can think of activity prediction problem as reconstruction of activity distribution and over a whole compound space. And we provide small number of compound activities, which will be obtained from our initial experiments in our workflow. And they are given to reconstruct the activities of a whole other compounds. So since the compound space is not defined spatially, like the images, it may be a little bit more difficult than the image reconstruction problem. But construction of compound space, such things has been quite successful for many tasks, such as designing the compound structures or properties. So we believe that we can handle that. So we are expecting that it be possible to do the same reconstruction tasks, like the image reconstructions on the lumped compound space. At this point, we had thought of two possible options to pick the compounds for initial experiments. One is using fixed set of compounds to perform experiments and testing, experiments and training the models. So I draw it like a grid-like structures. So we pick some compounds systematically and use them over and over again for different targets. It's the one way. And the other one is using a kind of flexible varying set of compounds for targets. So for a given target, we pick new set of compounds for initial experiments and use it for building the model for that target, and pick another set for another target. So actually, we had tried both options. So we build a model using fixed set of compounds or a flexible set of compounds. So at first, using the fixed compound set to provide the activity data. Actually, this method provides an easy way to manage initial experiments and also designing the AI model. So in the experiments, if the compound set is fixed, we can first buy or synthesize the compounds and just use them over and over again. So it can reduce actually quite amount of time for performing the initial experiments. And also first set gives the data as a form of a pixel expector, so which is one of the most standard data format for constructing AI models. So at first we designed workflows and models assuming the fixed set of compounds for initial experiments and model building. However, we found that fixing the compound set causes many problems in practical situations. So as a result, our final model is built for dealing with a flexible set of data. So I'll explain first about the preliminary model based on the assumption of the fixed compound set. So actually, we cannot find the experimental data set following this assumption. So there are no kind of such things that like a fixed set of compounds for used over and over for different targets. So we had to build a data set by using the docking simulations. So we collect about 2 million compounds and with 130,000 protein structures, actually the virtual pockets. It includes the virtual pockets from the Pocketon. The Pocketon pockets, so they are built by listing up the pocket-like structures by scanning over the surfaces of known protein structures. So it's a kind of virtual holes in the protein structures. So for the compounds and targets, from the 2 million compounds, we randomly selected hundreds of compounds for simulating a fixed compound set for model input, and docked them with all available pockets, 130,000 pockets. Also, the remaining compounds were randomly picked and docked into the pockets to generate the output data for training. And overall, we had performed about the docking simulations for almost 100 million compound pocket pairs. So from this data set, we built a model to predict docking score for ligands from a given activities of compounds. So although the model is quite simple, we didn't optimize it much, but it predicts the docking score. The docking scores quite well. So we have R-squared value around 0.4, so not that bad from the beginning performance. So we could conclude that if sufficient data is given, so it is possible to build AI model performing ligand activity-based prediction. However, as I mentioned earlier, transferring the model trained on the docking simulation for predicting experimental values was quite difficult. It is because we had used really fast docking simulations, which has really low accuracy. So the correlation between the docking scores and experimental measurements are weak, very weak. And also building on experimental activities for the fixed compound set from external data, experimental data is really difficult. Actually, there are no such data set exists. So we try to apply the missing value imputation method to make the full activity data for fixed set of compounds. But the errors are accumulating. So the accumulated errors from imputation and prediction degrades the performance further. So it's not good for the practical purpose. So we decided to build an AI model which is able to deal with a flexible set of activity values. Okay, so from now on, I'll explain about our main model, which is called meta-DTA. So we presented this work this year in ICLR workshop on machine learning for drug discovery. So the meta-DTA model does not rely on the structural information of the protein target. So we can apply it for the cases where the protein structure is not available. So we assume that no protein structure exists for this kind of task, and just to use the activity values, available activity values, to predict the activity of the target compound. And it can use the flexible set of compounds as input features, so the number and the order and which compounds is in or not is not important to build the model, build training and the inference, make an inference of the model. So we do not have to formulate the data in such a predefined way, so we can really use almost any kind of available data, external data. And most of all, the prediction performance is better than other structure-free models. We think that this is because of the use of the meta-learning methodologies. So I'll explain about the detail later. So I will briefly explain about the concepts of meta-learning we used. So meta-learning is also called as learning to learn. So in conventional machine learning, the objective is to figure out model parameters by training from data to perform a given task. So it's the basic machine learning. So in meta-learning, there are several tasks, similar tasks exist, and the objective is to figure out some meta-knowledge, which is then used to improve the performance on some distribution of tasks, including unseen ones. So as a result, for each task, the training can be much more easily and quickly done by using the learned meta-knowledge. So there are many different types of meta-knowledge proposed so far, such as the optimizer settings, hyperparameters, and initialization of some model parameters, or sharing some part of the models. Also, there are many different approaches proposed for learning meta-knowledge from the tasks. Okay, few-shot learning is one of the most representative application of the meta-learning. So it aims to solve each task by using only a small number of few-shot training data. So let me see. In this figure, the first example provides the information about 5 different classes with only one pictures for each class. So the first one looks like a bird, and the tank, dogs, a human, it's a piano. Then, so for given these images, then we try to predict the classes of new samples. So the first sample here looks like a dog, so it belongs to class 3. And the second one looks like a piano, so it will be class 5, belongs to class 5. So as you can see, in this case, the human can do this kind of task fairly easily, but typical deep learning methods usually cannot do this kind of tasks well. If only a small number of training samples are given, because the models have a huge number of parameters, and it may not be properly tuned with only a small number of samples. So meta-learning is an effective approach to deal with this few-shot learning problem by extracting and exploiting appropriate meta-knowledge from number of tasks, different tasks. So in the meta-learning, the training set for each task is usually called a context set or a support set. And the test set for each task is usually called the query set or target set. So we can think of the few-shot learning problem as the problem where, in the meta-learning problem, where only a few samples are given for a context set or a support set. Okay, as I mentioned earlier, there have been many approaches for meta-learning. So the most widely mentioned classification of meta-learning approaches classifies the methods into one of the optimization-based, model-based, and metric-based approaches. And the meta-detail models is closely related to a metric-based approach. So metric-based approach basically tries to build a similarity measure between data points. So this kind of building some kind of metric feature space, and predict value for some item based on the values for the similar items. Actually, this basic idea has been used in many different problems, such as nearest neighbor classification and collaborative predictions. And also, Ligand-based methods can be considered as a similar approach. So we use similar active ligands to make a prediction. But usually the ligand-based method, the similarity measure itself is usually defined manually in more chemical or biological way. So in this case of meta-learning, we try to build this kind of similarity measure from the data. And one example of metric-based meta-learning method is called prototypical networks. So recent benchmark showed that the prototypical networks had superior prediction performance than the other available approaches. So including one of the famous of optimization-based meta-learning method and multitask learning, so ensemble learning, other methods. Okay, and in fact, there was a few-shot learning method proposed earlier following the metric-based meta-learning approach. So basically, the model determines the similarities between the given compound set. So it corresponds to our initial experimental results and the given compound with this one. So the final prediction of the activities are calculated as a linear combination of the context activities, weighted by the similarities between the compounds. So it's it implements the really basic and intuitive ideas for the prediction models. And this is our meta-detail model architecture. The meta-detail model also tries to determine the similarities between the compounds. So similar to the previous work, the model is based on the attention mechanism to deal with the varying number of context compounds. So the attention mechanism basically compares the compounds and gather the information from the compounds getting more attention So attention itself can be considered as the similarity between compounds. So because the attention mechanism run on the kind of pairwise similarities, so the model can deal with the compound sets with a different size and organizations. And compared to the previous work, Metality has a more complex architecture to implement two different strategies. So one is treat the given context set compounds as a reference point for similarity-based prediction. And the other one is trying to extract target specifying features from the compounds. So we have implemented two different strategies into our model architecture. And then we noticed that the overall model architecture follows a general model framework, which is called attentive neural processes, proposed earlier from the Google. And for the attentive neural processes, the architecture consists of a deterministic path and latent path. And deterministic path corresponds to the first strategy, and the latent path corresponds to the second one. So this is the detailed view of the deterministic path. So here it looks quite similar to the previous works. So here the similarity between the support set compound and query set compound is evaluated by the model through the cross attention mechanism. And for each query ligand, the model collects appropriate contextual ligand information to generate features for predicting the activity. And the model consists of several layers doing this kind of process, so which makes it possible to learn really complex relationships between the compounds. And the latent paths of the model just summarize the several activities into a single representation. So we can consider the output of the latent path as a target specifying features derived from the set of activities. So here also attention mechanism is used to collect the information from varying set of compounds with known activities to summarize them to figure out one feature vector. And this is the final prediction module of the model. So it collects the outputs of both the deterministic path and the latent path, and use them to predict the activity of a given query compound. So it collects all the information and use them simultaneously to predict the activities. Okay, before I present the result of the experiments, I briefly explain why the meta-learning formulation helps to get better prediction model. So one difference between the conventional machine learning process and the meta-learning process is that the meta-learning uses splits of the training data to generate diverse training sets. So splits the training data into the support set and query set. So the different splits corresponds to different tasks. So we can generate a large number of tests from a given small number of samples. So for example, the conventional machine learning problem in the left-hand side, the figure tries to build a classifier which determines a given image is representing a kitten or not. So in this case, we have only 4 pictures to train the model. So because the number of training sample is so small, the model might be easily overfitted to some irrelevant features of the available pictures. And for the activity prediction problem, the situation is the same because usually the number of compounds for each protein target is not that large, so small. So typically less than hundreds, a few hundreds. So it's a usual situation. However, in meta-learning scenario, on the right-hand side of the picture, we generate different tasks by splitting the available data into context set and query set. So we used the context set to train the model for the task and try to evaluate and update model with the query set predictions. So you can see that with only 4 samples, there are 6 different ways to split the pictures evenly, 2 by 2. And if the number of data items increases, actually the theoretical possibility increases is about the order of the 2 to the n. So we can generate huge number of tasks by shuffling the available data points. And different split provides actually quite a different situation for training the models. So the model does not seriously overfit to some of the irrelevant features in the training data set samples. So actually we experience these behaviors during training our activity prediction models. Usually the conventional machine learning based models converges really fast. After only a few iterations passed, model converges and starts to overfit. So the generalized performance degrades at a point. But meta learning models usually converges quite stationary for much more larger number of iterations, does not overfit well. Okay, this will be the advantages of meta learning. Okay, here are from now on, I'll explain about the numerical experiments. We performed 2 experiments for the meta-DTA model. In the first one, we want to figure out how many few-shot samples are required to make accurate predictions. So we tested the prediction performance with respect to the number of few-shot samples given. And in the second one, we want to know that the model activity generalizes well for unknown targets if sufficient amount of data is given. So to test that, we control the diversity of the protein structures in the training set and measure the performance changes. And we use the binding DB data set for our test. So it contains over 1 million drug target binding affinities measured from experiments as a half per inhibition concentration values. Okay, and the settings for the models, we use 2 different types of meta-DTA models during the experiments. So we can apply the meta-DTA model in 2 different ways. So the first one is the inference mode. So after obtaining the initial experimental data, this mode does not use them to actually update the model parameters. So the model is applied without any fine-tuning, just use the data to make predictions. It just uses the data as input for the model. And the other one is the meta-DTA training mode. So it uses the given input data to fine-tune the parameters of the model. So in most of the cases, meta-DTA training mode has superior performance for the target task because it fine-tuned for the certain task, and because the fine-tuning itself does not requires much computational resources. So it will be more appropriate method for practical situations. We will use that, maybe, yeah. However, by examining the performance of the meta-DTA inference mode, we can estimate the generalization performance of the model for unknown novel targets. So we performed and reported the result of both modes. I'll explain it later with the results. And for the baseline models, we used 2 structure-free models for comparing the performances. So there's a deep DTA and mole trends. So these models use the protein sequences as features to predict the activities and by using the deep learning modules. So deep DTA is based on the convolutional neural networks, and the mole trends is based on more recently proposed transformer architecture. So This is the result of the first experiment. So here, x-axis represents the number of ligands in few-shot support set, so ranging from 10 to 100. And the bars are showing the prediction error measured by the mean absolute error and MA here. And as we can see, with increase of the number of few-shot data, model performance gets better for all cases. And the meta-DTA showed better performance than the baseline models, both the meta-DTA inference mode and training mode. And the performance of meta-DTA inference mode is usually better than the other baseline models. It is interesting because the baseline models actually uses the few-shot data to update the model. So it fine-tunes with the context data and also exploits additional protein sequences as features. So it is using much more information, but the performance is worse than the meta-DTA model in our experiments. And also we can see in the x-axis the random. Random means the ligands are randomly split into training and test data sets. So sometimes the model will predict the proper activities of the similar compounds. And the scaffold means we apply the scaffold split, so the training set does not contain the ligands similar to the ligands test set. So the overall performance degraded if we apply the scaffold splits. In fact, in current meta-DTA model, actually no special methods were employed to improve interscaffold generalizability. But it should be noted for this method to be used in the practical applications. And the second experiments try to measure the effect of the diversity of the training data. To test that, first we calculate the structural similarities between the proteins. So we collect experimental protein structures, and if there are no such structures available, we use the AlphaFold2 structures instead. And we calculate the TM score, and based on the by using this kind of similarity scores, we cluster the proteins into 32 groups, different groups. So by doing that, we expect that we can control the diversity of the protein structures by changing the number of clusters used in the training set. And this is the result of the second experiment. So the meta-DTA training mode and the two baseline methods showed good performance over the cases. So mainly because they use the context data to update their parameters. So by tuning and using the context data for training is really helpful to obtain the final performance. And the performance of DTA is actually degrades over, seems to depend more on the fine tuning of the data, as the performance becomes worse when more diverse protein information is given. And meta-DTA training mode showed the best performance throughout the cases, and it also improves with the diversity of the training data. So it seems that the model can effectively use the diverse protein structure data to improve its generalization performance. And yes, the meta-DTA inference mode showed the most interesting result here. So at first, where the diversity of the proteins are limited, the model's performance was really bad. So it actually cannot generalize well for the unseen targets. However, as the diversity of the training set increases, the performance gets better and better. And at some point, performance better than the baseline models. So it indicates that meta-DTA takes advantages from the diversity of the training data set to improve its generation performance on unseen targets. So the model is already prepared for some kind of unseen target proteins. So we can improve the performance further by fine-tuning. So far, I have shown the result of numerical experiments. So in fact, we applied meta-DTA models and the workflow for actual drug discovery project. So we pick two novel targets which does not have available structure data, protein structures, and we performed the initial experiments with about 300 compounds and obtained about 20 compounds with measured activities. So IC50 values ranging from 5 to 8. And build a model and screen the compounds. So from the initial experimental results, we build the meta-DTA model for each target and screen the compounds library to find novel active compound. And actually, we successfully found the novel active compound for the target. But we think that the accuracy is not that satisfying to screen a large number of compounds because mainly because of the difference of the data distribution in the training set and the actual experimental set. Usually the training set, training data usually has more abundant amount of active compounds, so it might lead many false positives. So our predicted activities are actually shifted to the active values. So for the future works, we think that careful curation and handling of the data should be done to improve practical performance. So there are many issues not solved in data handling, as well as the gap between trained data and experimental data distributions, mentioned in the previous page. So we have many out-of-range values. So many inactive values has actually not specified labels without having some specified values, just to have some ranges. So it is the activity is less than 4 or activities are greater than 8. So the most of the data has this kind of labels. And also there are different outcome types for the activities. So we use the IC50 values for the labels, but usually the experiments are done for the inhibition at certain concentration only. So if we build some method to deal with both types of this kind of various outcome types, it will be really helpful to improve the model performance. And we can think of another possible direction, which is curriculum learning. So by splitting the task in a specific way, it may be possible to train the model to learn more sophisticated features from data. So for example, to do this, we have to classify the tasks into a large number of possible tasks into easy one and challenging ones. And after learning for training the model for the easy tasks, the model will be gradually given more challenging tasks. So I think the scaffold split problem or such things can maybe the one good example will be the good example for this kind of curriculum learning. And also to eliminate the requirements for initial experiments, zero-shot learning based on appropriate protein features will be required. So actually we do not like the initial experiments at first. So adapting sequence or other protein specific features like baseline models can be a promising way to improve the workflow at the reverse stages. So here is a summary. We are focusing on the early drug discovery for novel targets. So few-shot meta-learning is a promising approach for such cases. And we presented a novel method called meta-DTA, and it also showed a promising result. Okay, thank you for listening. The paper and code for meta-DTA is available on the internet. And if you have more questions after the webinar is finished, please send emails to me or the first author of the paper anytime. And finally, we are open for collaborative research. Actually, we are widely open for the collaborations. So if you have any idea to use meta-learning based methods presented today, or to use the target and drug discovery platforms, Standigm, and Best, so please feel free to contact us through our homepage or just send me an email. So, and again, thank you very much for listening.
Well, thank you very much for that insightful presentation. I would like to invite our audience to continue sending their questions or comments right now using the questions window for this Q&A portion of the webinar. And I'd also like to draw their attention to the handouts module within the GoToWebinar control panel, where you'll find some additional documentation relating to today's presentation to download for some further reading. Now, I've already received some questions for you, Jiho, so let's get ourselves started with our first one here. It asks, what is the process after finding active compounds through meta-DTA? They further say that maybe there are still no accurate receptor models for analyzing structure-activity relationships at that point, but at some point, the structure-based analysis will be essential.
Yes, it is a really good point. And basically, we could apply the ligand-based approach to find the additional active molecules. Standigm-Based platform provides the module for the ligand-based methods, and it can be used separately for such cases. As mentioned here, in the later stages of drug discovery, the accurate structural model is really helpful to drive compound structures for optimizing properties through the SAR analysis. We are also thinking of that. So if we obtain promising active compounds from meta-DTA workflow, while the ligand-based methods are first applied to find the more active compounds. The effort for obtaining accurate structures of proteins will be simultaneously done. In that cases, the active compounds found from meta-DTA workflow will be helpful to determine the holoform structures, which is the protein structure induced from the binding of the certain active compound.
All right, excellent. Thank you very much for that very detailed answer. I appreciate it. The next question I have for you would like to know, how do you select the compounds for initial experiments? And how many would you say initial experiments are required to build an accurate model?
Okay, good question. So the first question is about the way to pick the compounds for the very first experiments. There are a few things we considered. So the first one is it may be better to use kind of popular compounds which have many activity measurements for diverse protein targets. Then we expected that these ligands will serve as something like a pivot point, which helps the model to distinguish the targets in training and data and the target in consideration. And another thing we consider is the coverage of the compounds. As we saw in the numerical experiments, the accuracy of the protein gets worse for the compounds having different scaffolds. So the compounds in initial experiments that should be similar to the compounds to be screened. So one possible way to achieve this is applying clustering for commercial library and pick representative compounds from that. There would be many possible heuristics to choose the compounds, such as using the uncertainty estimations or inference with the part of compounds and so on. In our practical test, we pick the compounds based on the popularity and add some compounds randomly selected from the compound library to improve coverage. So, but yes, there are lots of other possible methods to do that. maybe we will figure out better ways later. And the second, okay, the number of initial experiments. At that point, we have some answers in our paper in the supplementary section. So we tested the metadata model in two different modes, so metadata inference mode and the training mode. And for the metadata inference mode, there will be there should be about 100 future samples to the model performance actually greater than 0.5 as the R scale values, and then the metadata training mode we required about 50 initialization examples.
Excellent. That's very good. Thank you very much for that answer and for all of the answers. However, we have reached the end of the Q&A portion of the webinar today. Now, if we couldn't attend to your questions, though, the team at Standigm will follow up with you. Or if you have further questions, you can direct them to the email address that's up on your screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it will help us to improve our webinars. Now, I've also sent you a link in the chat box, and with this link, you'll be able to view the recording of this event on this page, and you can also share this link with your colleagues when they register for the recording here as well. So I encourage you to do that. Now, please join me once more in thanking our speaker for his wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thanks for coming.
Okay, thank you for listening.